And people keep alluding to this. I hope you like it. Yeah, today I'm going to be talking about using the same techniques that Dan talked about and the resources that Jim talked about, and sort of a real-life example that we've been working on in Mimulus catatus, or yellow monkey flower. If you're interested, there's probably some around here, so keep an eye out for charismatic little flowers and bright yellow. So today I'm going to put this into a context for assisted gene flow and what we need to get from point A to point B using these genetic methods. So the first thing you need to know is understanding how these populations are interacting with each other across space. You then have to know your environment. That's probably the most important thing that you need for this. We have to figure out which of the alleles that are popping out are the most likely ones that we should be looking into as targets. And then we have to figure out which populations need them the most. And so those are sort of the 4 steps I'm going to take us through today and how we can use these methods to answer those questions. So let's get that first one, understanding how populations interact. Okay, well, question 1, what populations are we looking at? So this is Mimulus, this is that nice little flower I talked about. We have 327 individuals across 110 populations from California and Oregon, which you can see here. This sort of covers the breadth of what we would look for the heterogeneous environmental landscape that we see across California. This is what they look like. You can see that we don't have like a simple transect. One gets slightly harder. It's this really multifaceted environmental landscape that we can like pull little bits from. And Mimulus is good for this because it grows everywhere, everywhere. Whether it's wet or cold or hot or really shady, right next to the ocean, it grows everywhere. So we can really sample all of that as best as possible. Then we have to figure out what they're doing with each other, or rather how much a population that's right next to one is similar to one that's next to it. So this is showing ancestry estimation projected onto geographic space. It's basically a method that we use to figure out if we had to divide this whole population into groups based off of genetic similarity, how many groups would there be and where would they be? And it seems to be really latitudinally correlated, which I'm not surprised for the West Coast. So we have Oregon in blue. This is supposed to be green for Northern California, and then for Southern California we have it at the bottom in red. Remember those colors, I'm going to be using them for the rest of the talk. When we look at genetic variation as a whole, which is this PCA you can see here on the side, you see that when we're looking at that genetic variation, we see that same sort of latitudinal line along the axis that explains most of the differences genetically between our populations, where we're seeing that same latitudinal change from right to left here with our colors. And I'm not going to be talking about it today, but this is due in large part to something called isolation by distance, which is if you're closer to something else, you're more likely to have gene flow already happening, so you're more likely to be similar to one versus the other. So we use that kind of information to figure out how far away are things that matter and what sort of similarity are we looking for between the two. And we compare that to environment. I think this is the most important thing from this. Environment is really difficult to characterize, especially when it comes to drought. When we think of drought, we're like, oh, the water's gone. But drought is way more complex than that, and is really a multifaceted collection of different combinations of variables that can make it stressful in different ways in different places. So to capture that, we have to characterize it also in a multifaceted manner. So the first thing that we used was the World Clim database. This is sort of the poster child for looking at environmental differences, but this largely colors just precipitation and temperature data, and like I just said, there's way more than that. So we also have combined this with the BCM model that we just heard about from Jim. So that's giving us not just temperature and precipitation, but all the hydrological inputs and outputs from our system so that we can characterize that on a fine scale. For the stuff that I'm going to show you today, it's a combination of this, but it's pared down so that we're not getting too much correlation between the two. Here in California, the coldest time of the year and the wettest time of the year are at the same time, so that kind of stuff is really correlated with one another. So I end up with the final 18 variables that we're not going to talk through that try to get at that sort of differences between the two. And we use this as an input into our overall method. So we took all those natural populations, so 110 populations, and we put them together in the same conditions, and we grew them in a common garden. We got full genome sequencing for all of them, and then we start funneling it into 2 different things. So I'm going to be calling it Genotype Environment Association Analyses, or GEA, but this is that environmental GWAS that we just heard about. So this is a correlative method. We're looking for clinal associations between environmental variables and the genetics of the populations that live in those environments. Today I'm going to be talking about two different methods that you can use for this. The first is redundancy analysis, or RDA. This is a multivariate way of looking at it. So when I'm talking about our RDA data, you can think about it as, I call it air quotes environment as a whole. And then we compare this with our latent factor mix model, or our LFMM, which is a univariate method. So instead of looking at it as a whole, comparing this to something like population structure, we can say, what about snow? Or what about evapotranspiration? What about these individual bits? So we did this for the entire range I just showed you, and we broke it down into regional models as well, based off of those segmentations. That ended up being really important, and you'll see why in a little bit. But we did that for both of these, and then in conjunction with that, we also did that trait GWAS as well. So because we grew all these in the same location, under the same conditions, we were able to measure lots of different traits, and then use that also as an association. So then connect that phenotype back to the differences we see with genetic correlation with environment. We did this in a couple ways that I'm not going to talk about. But combined together, that gives us a short list of putatively adaptive genes that could be useful for local adaptation to drought, instead of the 430 million base pairs that we started with. So I'm going to show you some of the outputs from that. So I'm going to show you the redundancy analysis. And if you ever get lost for which one I'm talking about, I have the symbols on the bottom. Redundancy analysis is that air quotes environment. So we're looking at everything at once. So I ran a bunch of these models. The first looks at everything. So we're looking at population structure, looking at geographic location, and we're looking at the environment, testing how much variance can we attribute to all of it. Then we're running it just looking at the environment and canceling out the other two. Then I have two more that do the same thing with each. So now we have a model that tests just for each of these particular items, especially in populations where we see latitude is correlated with something. Latitude may also be correlated with the environmental variables. You need to cancel that out as much as you can. What you get out is something that looks like this. And the most important thing to know is what's in that red box there, which is that when we're looking at environment on our full range data, we can attribute 10% of the explainable variance to environment alone. So we're able to pull out just what's important for that, although there is lots of interaction between these things, and that sort of gets confounded into a sort of a mixture variable. Now if I look at that only individually on those regions, that number minimum doubles and then triples in Northern California. So that's showing us that that major population structure overlay on the full range is obscuring a lot of the stuff that might be important in one region versus another. So it is really important to think about the environment that you're working on for these kinds of studies so you can tailor it to the unique different discrepancies that you see across the full range. Which I'm alluding to here, but I'll show, which is for these different regions, we segregated them based off of genetics. The next question is, are they different environmentally as well? What you can see is that these two ellipsoids, the red and blue for Southern California and Oregon respectively, they sort of just kiss, they don't really interact. So those are really fully discrete regions from one another. Northern California acts as a bit of a bridge between the two, which is not surprising. But this tells us not only do we see genetic clusters, but they tend to be environmentally distinct, and then also end up resulting in different selective pressures. So this is our LFMM data. This is letting us look at each individual variable. What's exciting about this method is it gives you estimations, at least on our side, of what variables might be most important in these regions. What you're seeing here is based off of the number of genes that are involved in these various processes, as well as how significant the hits we're finding. What you can see from this is that for each, our full range and all of our different submodels, the selective pressures of most importance for the way we're defining it are distinct between the two. So something that's adapting to higher elevation in the north is not going to be the same thing that's adapting to really intense drought in our south section. So that gives us a bit more information about the environment that we're asking questions about. So then we get to the meat of everything, which is, okay, well, what matters from this different discrepancy? So this is one of those Manhattan plots you just saw. It's not as beautiful as Dan's, unfortunately, but I've highlighted a couple of peaks that we're going to use as a through line for this as we continue. The first is phytochrome E, which is our highest peak, although our highest bits are a bit more sparse than the rest of the peak down lower. And then we have TAF5 in the corner here. Both of these are genes that are involved in a flowering time pathway, which is something that is potentially important to drought. In particular, it has to do with drought escape, which is a live fast, die young approach where plants try to flower as quickly as possible. That way they can sort of outpace the impending drought that's on its way. So phytochrome E is involved in this photoperiod pathway where it's looking at red light and it's triggering flowering. And then TAF5 is a more generalized transcription factor that most importantly affects FLC. What are those genes? You don't need to know, but you need to know that they end up doing the same thing, which is that it results in some sort of modulation of flowering time. Now we did all these different models, and what is exciting about that is we can then overlap them and increase our trust and our confidence that these are real things that matter. So we're going from big lists to smaller to smaller to smaller. So let's look at that overlap. So this is looking at, of the outliers that we get for each of our models for our RDA, most of it tends to be distinct. So you only find it in one environment, which makes sense. We're seeing different environments, we're seeing different selective pressures, so it's selecting on different genes. So we can pinpoint things that way. But it also begs the question, okay, well, what is it that's important across more than one? And is that something that we can use to our advantage? Surprise, the one that overlaps with Southern California is phytochrome E. That's the biggest one that's coming out from this. And when we look at the overlap with our Oregon, we also get TAF5 again. So that's increasing our confidence that this is something that's important and important on a wider range as well. This next slide is busy. You don't need to worry about the busyness. I'll point out the guys that matter. And that's the familiar face that we're seeing right here. We see phytochrome E yet again. So this is looking at our LFMN data. You can see it's univariate based off of the environmental variables at the bottom. And what's exciting about this is one of our biggest ones that matters the most for our full range data is evaporation, and we are hitting hits in Phi E for our variable of importance. So that lets us sort of narrow that in as much as possible. And I have a couple others that are pointed out that are all fitting into that exact same pathway. So we have an idea that that glowing, that flowering time really does matter quite a bit. And then we can compare that back to the thing I've been ignoring this whole talk, which is our trait changes. So looking back at, does this actually result in some kind of phenotype that can do something for us? Now, if your first question is, well, Phi E isn't here, that's a great question. We didn't get that as one of our outliers, but we did get something that's directly related to it in that same pathway. So this is suppressive frigida 4. It's involved here. It directly affects FLC in the same way, well, not in the exact same way, but similar to what we see with TAF5. And we see that it's significantly associated with time to flower, and that it overlaps with evaporation specifically. So we're starting to get all these through lines that are pointing to a smaller and smaller list of what might be an important allele that we should look at when we move forward. And this is just one of many, many, many examples that we get out of this. And then that takes us to our last little bit, which is identifying which populations are most at risk. So there's a bunch of different ways that people have talked about it so far. All of those are valid. You should do all of them. I actually think the one I'm showing you is not enough to do without also doing the others. But using our genetic data, the way we generally do this is by looking at something called genetic offset, which is a proxy for an estimation between what the difference is between the allele frequency you have now and the allele frequency you need to have to survive in the future. So I'm doing gradient forest modeling for this. It's a machine learning method. So you basically train a model on that current environmental data set that we already have, and then we take projected future data sets. For me, I got that from World Climb. I have 3 low, medium, and high stress projections for what our future could look like. And then it lets us look to say, okay, this population's doing good now. Will it be doing good later? And what can we do about that? So this is just a big summary graph of what that might look like in comparison to, yet again, evaporation. You can see that, yes, as stress gets worse, you see there's more genetic offset. We can also see that as there's less evaporation and thus likely less water to be evaporated, that genetic offset does get worse. We can look at those outliers at each of our levels and pull them out, and this gives us a list of which populations are there's the biggest discrepancy between where they are and where they need to be. So it's not just how close are things, or is there gene flow that's already happening, but helps us project things into a future distance and then overlap that with our other data as well. So with all of that together, we know what our populations are doing, what they're inhabiting, what it's like to live in one of those places, what alleles might be the most important thing for them to adapt to the places that they live, and which ones are most likely to be not able to do that as it continues forward. So through this pipeline, it helps you go from A to B, B being effective gene. And that's it. A bit of shameless self-promotion. Yay, I'm about to finish my PhD, which means I'm on the job market. So if you want someone to do this for you, or a friend of yours, talk to me.